Okay, thank you very much. Right, so the plan for today is to just recap what we've talked about in the previous two webinars, and then I'll explain why at Horizon we've moved towards a haploid-based model to do our genome editing in a more scalable way, and what some of the advantages are of doing that. I'll then show you some of the data that we've generated during the course of the 1,000+ knockout experiments that we've performed. And then I'll show you where we've taken this model in terms of the modifications that we're now engineering, the kind of efficiencies that we see, and the approach that we've taken to doing gene tagging in a scalable way. And for those of you that hang out with me till the end, I'll also tell you about our upcoming website launch and how you can get 10% off your next order of X-Man cells. Okay. So just to remind you, in the first webinar, I made the case that genome editing is definitely the most robust way to translate genomic information now into translational outcomes through truly understanding the role that genes and mutations play in driving disease, for example. And we explored how you could use CRISPR to introduce knockouts through the introduction of double-strand breaks and subsequent frame shift mutations, or knockins through homology-directed repair and insertion of exogenous sequence. And last week, I spent quite a lot of time looking at the key considerations for genome editing, namely the choice of cell line itself and whether it's suitable, how your gene target and the modification of that target will affect the viability and ultimately the success of your experiments, how guide choice can allow you to balance specificity and efficiency, and how the position itself will depend on the modification that you want to make. I also highlighted ways in which you could design your donor such you could maximize your efficiency and minimize the chances of it being recut once it's integrated. And then finally, we looked at screening strategies and how validation is an important final step for generating any modified cell line. So with this in mind, let's move on to today's topic, which is the kind of modifications that you can make in mammalian cell lines. Now, last week I highlighted some of the key challenges to generating any modified cell line. But notwithstanding the efficiencies that you can achieve with CRISPR and also the ease of design, challenges do remain. And a big one of these, at least for us at Horizon, where we're trying to do potentially tens, if not hundreds, of genome editing experiments each month, is the fact that every cell line, or almost every cell line I should say, has multiple copies of your gene of interest. And what this means is that if you want to knock out your gene using the frame shift mutation approach, for example, you've got to confirm once the CRISPR-Cas9 disruptions have occurred that every copy of your gene contains a frame shift mutation. And there isn't really a quick way to do this. At Horizon, we would amplify the target locus. We would subclone that into a plasmid, for example, a topo vector, which we would then use to transform competent bacteria, and we'd then subsequently sequence the mini-preps produced from those bacteria to deconvolute the alleles, if you like. And this, if you're trying to do tens of genome experiments at a time, can become a significant bottleneck. It's not possible to simply sequence the target site, as the random nature of NHEJ-based repair means that you'll often get different modifications in each allele. And as a consequence, if you simply sequence the target site, you would get messy Sanger trace. And you can see an example of that here on this side, where we've aimed to disrupt the MAPK3 gene in the A375 cell line. And this is a cell line that contains 3 copies of this gene. And if we target the MAPK3 gene, we'll actually get, in this particular clone, we have 3 different types of modification, all of which ultimately turned out to be frame shift mutations, but we wouldn't have known this had we not subcloned the target site. So how do we get around this? Well, actually, the approach that we've taken is to move towards a haploid model. Now, this haploid system and this sort of approach to doing a loss of function analyses in haploid cells was originally pioneered by this chap here, Thymme Brummelkamp. So the first haploid cell line to be derived was the KBM7 cell line, and this was derived about 25 years ago from a chronic myeloid leukemia patient, and it's actually a near haploid cell line. So it contains 2 copies of chromosome 8 and is disomic for parts of chromosome 15. And despite its uniqueness, it's sat relatively unused for 25 years until Tyne, who realized that from his experience of doing siRNA-based knockout screens and was acutely aware of the limitations of that approach, realized that if he combined this cell line with a promoter trapping-based approach, he could have a very robust methodology for doing loss-of-function analyses at a genome-wide scale. And he used this very effectively to determine pathogen host factors. But the same strength, this fact that you only have a single copy of each gene and therefore you can very efficiently knock out genes in this cell line, also lends itself to doing CRISPR genome editing. As not only can you more efficiently target in that cell line, so for example you only have to modify a single copy rather than multiple copies, but it also releases this validation bottleneck, as you can straight away sequence from the genomic DNA and ascertain the nature of the modification in your particular clone. And so to this end, Horizon have actually developed a panel of near haploid and haploid cells. So we began with the KBM7 cell line, but from this we derived our HAP1 cell line. And this again is a near haploid cell line. It contains that disomic portion of chromosome 15, but it's lost that second copy of chromosome 8. But importantly, for the purposes of or for the process of genome editing, it's an adherent cell line, and so it lends itself to that process of single cell dilution and identifying clones. And finally, and I'll touch on a little more on this later on in the talk, we've actually since generated a fully haploid cell line from which we've excised that final disomic fragment to give us a fully haploid EHAP cell line. And so what this means is that we now have an extremely efficient and easy to use and robust genome editing system in the form of CRISPR, but we also have a highly efficient model in which we can generate knockouts. And so if we combine these two, we can generate a high number of knockouts in a very scalable and cost-effective way. And actually, to date, we've generated over 1,500 knockout cell lines using this cell line. We've built up a significant body of data around the cell line, so we know what is and isn't expressed from our RNA-seq data. We've also done essentiality profiling to find out what can and can't be knocked out. And as a consequence, we can now take pretty much any gene of interest, and provided it's not essential, we can knock it out in 10 weeks, and our academic pricing is for under $2,000. So by contrast to diploid cell lines, where you have this very labor-intensive validation step, this model really does lend itself to generating a lot of knockouts in a very rapid way. And this is really what's enabled us to generate quite extensive collections of knockout cell lines, and you can either group these together into gene collections, for example, so we have a knockout for every non-essential kinase in this cell line, or into pathway collections, so for example, mTOR signaling. So from the process of generating these knockout cell lines, there's been a certain number of observations that we can make, and I thought it might be interesting to share these with you now. So first of all, what we found is that for the large part, this is a highly efficient system in which to do genome editing. So you'll see that the high proportion of our experiments, we observe a greater than 40% editing efficiency. And in this system, the way in which we measure editing efficiency is that for every targeting experiment, we plate out 12 clones and assess these for disruption at the target site. And if we observe any disruption, then that's considered a positive. So for example, 50% targeting efficiency in this system would be 6 out of 12 clones having undergone some kind of on-target disruption event. As we might expect, the mutations tend to cluster around the site of the double-strand break, which is 3 bases to the 5' of the PAM site. But what's interesting is that the the mutation we see with the highest frequency is a 1 base insertion. Although having said that, we tend to observe deletions with a higher frequency than we do observe insertions. Now, one thing we wanted to understand was the likelihood of off-target disruptions in this system. And so to do that, we took 10 clones in which we'd already confirmed the on-target disruption, and then took the 10 most likely predicted off-target sites for the guides used to generate those clones, and looked for evidence of disruption at those off-target sites. And in no case did we find any off-target disruptions. And so just to summarize this section, what we found with targeting in HAP1 is that it's a highly efficient system. We've seen that our mutations cluster around the PAM-3 site, and that generally speaking deletions are favored over insertions, although the 1 base insertion is the most common mutational event in the system. And we believe that off-target editing represents a minor issue. So where do we go next with this system? And this is what I want to spend the rest of this talk talking about. What are the kind of modifications that we can make in this system? What are the efficiencies that we observe? And how do we go about doing it? Well, there are a number of different modifications that are desirable. Not everyone wants to do loss of function analyses, especially if you're working on essential genes, for example. And so you may want to introduce a point mutation or correct a mutation, for example. Translocations and fusions, or indeed chromosomal deletions are desirable. And also of interest is the ability to tag genes at the endogenous level. And these can be used for expression analysis, subcellular localization, or indeed it's just a wonderful way to work with a gene or a protein for which you don't have a good antibody. So the first one I want to talk about is the introduction of point mutations. So as I've mentioned previously, the way in which this works is that you would target your CRISPR-Cas9 to a target site in the genome, and you would, where it would introduce a double-strand break, and this would be done in the presence of a homologous donor sequence. And then in some instances, you'll observe repair of that double-strand break via the homology-directed repair pathway. And in this manner, you can introduce your single base changes. And so I showed this example last week. This is where we've introduced an L858R mutation into the EGFR gene. And critically, we did this at the same time as introducing a SPE1 site, which allowed us to screen for those clones into which the donor had been integrated. And you can see here clones 5 and 10 are susceptible to SPE1 digestion, which indicates that they have integrated the donor. And so of the 23 clones we screened, we observed 2, which is approximately a targeting efficiency of about 8%. We have also been interested in introducing translocations and fusions, in introducing chromosomal deletions in the system. And a big driver behind this is the fact that the HAP1 cell line is not quite haploid. So it still contains this chromosomal, this disomic chromosome 15 portion. And for it it would be really useful to us to actually remove this, as it then becomes a much more powerful tool, not only for generating knockout cell lines, but also for the purposes of doing knockout screens, for example, as we don't then have any part of the genome that's present in a non-haploid state. And so the way in which we set about doing this was to design guides both upstream and downstream of this disomic fragment. We designed multiple guides and tested them in various combinations. And also we then designed PCRs flanking the region that we aim to excise, and we designed primers rather, and used PCR to look for evidence of that region being cut out, and then these two fragments coming back together to give us a PCR product. And you can see here individually the guides by themselves, no PCR products, but really it was only when we combined guides 1 and 3 did we observe excision of the interceding portion. And so we took this pool population forward, we plated out 400 clones, and of those 400, we observed 5 in which we'd excised this 30 megabase fragment. And you can read the complete experiment in this publication here at the bottom. So this is again a 1% targeting efficiency in this particular experiment. Further to this, we could validate these clones not only at the genomic level, but again by the spectral karyotyping. And so you can now see chromosome 19 is complete without that disomic fragment. Next up we wanted to experiment with introducing translocations. So there are a number of translocations that are associated with cancer. Perhaps one of the best known is the EML4 ALK translocation. And so it was desirable to us to be able to recapitulate these in a model system for the same reasons that we'd want to introduce disease-associated point mutations, for example, so that we could study the role of these translocations in driving disease and in phenotypic analyses. So the way in which we did this was to design a guide in the breakpoints that we wish to introduce and at the sites of translocation. And you can see a schematic of that here. And further to this, we then designed primer pairs such that we could screen for clones in which the translocation had occurred. So we had, in this case, we're trying to introduce the CD74 ROS1 fusion. And so we have primers in CD74, primers in ROS1, and we can look for evidence of the translocation in our clones. On this occasion, we plated out approximately 200 clones, and of those we screened, we found 2 that were positive for the presence of the translocation. And this really highlights the advantage of using the 2 different PCR primer pairs. You can just about see the positive clones coming through in our CD74 ROS1 screening PCR, but it's very obvious once we use the alternative primer pairs. And so 2 from 200, we're looking at about 1% of our clones are testing positive for this particular fusion event. Again, we would then want to validate these, and we did this at both the genomic level. So in our 2 clones, we observed slightly different breakpoints. So you can see this here on the left-hand side in the bottom left-hand corner. But in both cases, the breaks occurred pretty much as predicted to where our guides cut. And we can also confirm the expression of the fusion transcript using RT-PCR, and you can see that in the bottom right-hand corner. So the final thing I want to talk about is the introduction of endogenous gene tags. And the reason I think this is interesting is because we've taken a slightly different approach to doing gene tagging. And the reason for this is because we want to be able to, in much the same way that we do with knockouts, we want to be able to generate tens, if not hundreds, of endogenously tagged cell lines. But unfortunately, the conventional approach to doing gene tagging doesn't really lend itself to doing that. So generally, or rather conventionally, you would do this by designing a homologous donor sequence that is your tag flanked by 2 homology arms to the specific site into which you want to integrate your tag. You then introduce your cut, and in some instances you'll get integration of your tag at that site. Now, the downside to doing this is first of all, it's relatively low efficiency, and further to this, every single targeting experiment that you want to do will require a specific donor template to be synthesized. So if, for example, you want to create an N and a C terminal tag, or if you want to try out different types of tags, you would need to synthesize a donor construct in every single case. And this doesn't really lend itself to making a lot of cell lines. So instead, we have adapted an approach that was first outlined in this particular publication here. And the way in which this works is that rather than using a homologous donor template, we use what we call a self-cleaving or self-releasing tag. And this is a plasmid in which you have your tag flanked by 2 guide target sites, and on the same plasmid you have your guide that would recruit Cas9 to those target sites being expressed off a U6 promoter. And so if you co-transfect your cells with your Cas9, your guide to target your particular gene of interest, and then your self-cleaving tag donor, then not only will you get cleavage of your target gene, but you'll get release of this tag from that donor. And what we hoped is that in some cases we'll get simply integration of that tag via non-homologous end joining based ligation. Now, we hypothesized that this would occur relatively imprecisely, but we hoped that it would occur with high efficiency. And indeed, when we tested this, we found that this seemed to be the case. So what we're looking at here is evidence of the tag being incorporated in the correct orientation into 7 different genes. Sorry. And so we've designed a primer in our exon of our gene of interest, and then a reverse primer in our tag. And in all but one case, we observe integration of the reporter cassette in the correct orientation. And if we then take these forward for clonal analysis, we do observe a range of different targeting efficiencies, and you can see that here, ranging from as low as nothing to as high as 21% editing efficiency. But we do seem to be able to achieve quite reasonable targeting efficiencies using this approach. But one thing that concerned us is that of all the clones we tested, we only observed one in-frame cassette integration. And actually, the reason for this was that, contrary to our expectations, actually the tag goes in quite precisely. And so we hadn't really anticipated this. We hadn't taken into account that the tag would actually go in at the breakpoints as it would be cleaved from the donor, and as the gene would be cleaved by the Cas9 itself. And as a consequence, we hadn't adapted the frame of the donor to take this into account. And so it was only actually those events where the tag had incorporated in an imprecise manner did we then observe in-frame insertion of our tag of interest. And so we very successfully incorporated our tag into multiple different genes, but because we hadn't anticipated the precision of this system, as a consequence, we generated a lot of clones that we couldn't then use. And so since then, we've actually created a suite of self-cleaving donors to account for all possible frames, and we would then use the most appropriate frame donor for each particular targeting event. Now, importantly, beyond just inserting the tag, you also want to make sure that it's inserted in only a single instance. And we would do this using digital PCR at Horizon, whereby we would ratio the tag itself against our gene of interest. And so you can see here we're using copy number analysis, and we have just a single copy of insertion of the nanolook tag into both the ID1 gene and also the DACT1 gene. And here we have a positive control, a nanolook line that we've created in the past in our HCT116 background, and here's our negative control on the right-hand side. Further to this, we also want to confirm that these cell lines behave as we'd expect, that they actually provide a useful biological model. And so one example of that is that we would expect DACT1 expression to be upregulated in response to stimulation with activin A. And we can use nanolook expression in the nanolook tagline as a readout for this. And so you can see after both 4-hour stimulation and after 24-hour stimulation, we observe upregulation in nanolook expression, which is a readout for the DACT1. So just to summarize this section, we now, in our HAP1 model, are able to reproducibly achieve targeting efficiencies of greater than 40%, which of course lends itself to generating knockouts very efficiently and in the kind of numbers that allow us to achieve an economy of scale. But further to this, we can also generate other modifications at the kind of efficiencies that are acceptable to us if we're going to do this cost effectively as well. So for example, point mutations, we've seen efficiencies of 8%. For large chromosomal deletions and for chromosomal translocations, efficiencies at 1%, and for our NHEJ-based ligation gene tagging approach, up to 21% tagging efficiency. And so I think this highlights that this combination of CRISPR and the haploid background really does lend itself to both simple and complex genomic modifications. So we've had 3 webinars, and I just want to kind of wrap up now. I hope from these webinars you've learned that, yes, CRISPR-Cas9 genome editing can be easy. It can be easy to design. It can be efficient. It is, generally speaking, widely applicable, and it's quite flexible as well. But I also hope in webinar 2, I highlighted some of the challenges that remain, not least that not every cell line is easy to target, not every guide is active, and regardless of how efficient a system you're working in, it's almost always a labor-intensive process to do genome editing, and it won't always be successful. And so I'd just like to finish by highlighting some of the ways that Horizon could potentially help you in your research. And this is really to say that actually it's very possible that we've already done the experiment that you're planning to do already. To date, we have generated over 1,500 different knockout cell lines. These have all been verified by Sanger sequencing. And for those of you that are concerned about the potential for off-target cleavage, we have generated multiple independent clones which are available for every gene, as the likelihood of those off-target events occurring in independent clones is very low. Now, importantly, it's fair to say that the HAP1 model is not going to be an appropriate system or the most desirable system for every person planning on doing genome editing. And so for this reason, we are able to supply the guide RNA used to generate that cell line in combination with the cell line. So you can get up and running with the HAP1 knockout, and you can take the guide that we've used and use that to generate a knockout, for example, in your cell line background of choice. And as with all of our isogenic modified cell lines, they're supplied with a wild type control such that you can really attribute any phenotypic observations to the genomic modification made in that cell line. So these cells sell at $990 per cell line for academics, and slightly more for industry pricing. But this isn't just all we do. We do genome editing on demand in our haploid cell line, and this is by far the most cost-effective way to get to loss and gain of function models. We can do the knockouts on demand or genomic deletions, as well as some of the modifications I've talked about today, the knockins, translocations, and tags. We do a fully custom cell line engineering service in diploid or any cell line of choice with any particular modification. And as discussed, this creates some challenges around, or rather, this can be a more labor-intensive process, and so there's a bit more of an associated price tag, but that is available as well. We've recently launched an induced pluripotent stem cell gene editing service, in which we would take your iPSC and introduce whichever modification you desired in anywhere from 12 to 18 weeks. And we're not just confined to cell lines, so we do in vivo genome editing as well. We already have generated many mouse and rat models, and you can find out more about those on our website. We do sell microinjection ready guide RNAs, and we're able to do custom in vivo gene editing.